think people just keep on coming in.
Yes.
So this lecture is going to be recorded, right?
Yes, doctor.
Okay, wonderful. I'll get started now. Okay. Hi everyone. Assalamu alaikum again. So let's get started on this. It's supposed to be a lecture, but let's view it more as a brief talk. Let's say let's talk about it. It's better to view it in that particular context. So let me just. So would you like me to start the recording, or are you going to do this on on your computer, on the cloud, or whichever one? If you just let me know your preference. Okay, starting now. Sorry. So yes. So we talked really briefly in my previous discussion with you about a brief introduction to sperm defects, and just that, considering the questions that I've been getting over the past week or so from a lot of you. Look, just focus on the basics. I don't expect details from this. This is quite advanced stuff relative to your level. Just approach it with the thinking that you need to be aware of certain things. Okay, so if you approach it that way, then you'll be able to kind of get it onto a brief perspective of what is expected to be kind of known from you. And the same thing with this one. Okay, this isn't something that your finals are going to be dependent on. I mean, yes, you will get questions from this, but it'll only be very basic stuff, and use the slides as your guide. Okay, I wouldn't expect you to know things outside of what's going to be discussed in this lecture. So what we'll be talking about for this session is a brief overview of what assisted reproductive technology is. Okay, I briefly mentioned to you that these were treatments, let's say, for fertility. And before we talk about that any further, let's just have a quick refresher of the whole purpose of having these particular technologies in the first place. So you see, the main reason why we're even talking about this, the main reason why we're even considering this is, of course, human infertility. Now, as we talked about briefly, the actual definition, if you want to go about it, as far as the WHO is concerned, as far as any characterization is concerned, actually, is that it's characterized as a disease. Okay, so it's not a taboo kind of condition. It is an actual disease that afflicts mankind, as far as the WHO is concerned as well. So globally, it's recognized as a condition people should seek help for. It's essentially going for a certain period of time of having regular, unprotected sexual intercourse and not being able to conceive a natural birth, or a natural child anyway. Birth that comes under a different definition. But the amount of time that one needs to do this, it varies, okay, between country to country and between definition to definition. As far as the WHO is concerned, that's what we're going to stick to, because that seems to be the standard throughout, as it were. So it's about 12 to 24 months or more of regular unprotected sexual intercourse. Okay, so this is also something to be very careful of, especially culturally speaking. A couple gets married, they're not able to conceive within the first month, and everyone's like, oh God, there's some sort of problems happening. Well, it doesn't really quite work that way. Okay, there's many other things that need to be thought about as well. There's lifestyle issues. There's also timing issues, according to the menstrual cycle. We're talking about, you guys know briefly about the fertility window as well. So, it has to be this specific time frame. Or at least a specific time frame as far as the WHO is concerned. It's about 12 to 24 months. Now, again, a lot of people don't realize this, but infertility is actually quite a big issue, and it's a growing issue as well. And there's lots of facts and figures which collectively are actually quite worrying when you look at them together. We'll go through a few more towards the end of this talk, which make the masses quite even more worrying as well. But globally, infertility seems to affect about 1 in 7 couples. So 1 out of every 7 couples at least will be infertile. It's thought that a higher proportion of these, as we discussed in the previous lecture, are going to be subfertile. So not outright infertile, but will have difficulty in conceiving. It won't be as straightforward as it is for many other people. And particularly in men, Its subfertility seems to affect about 1 in 5 males worldwide. So fertility issues are increasingly becoming of concern, particularly considering that it seems like a catch-22 is that, well, you could say the population of the world is actually increasing, but ironically, the more the population increases, the more issues with fertility people seem to be having, and again, multifactorial, because there's not one straightforward cause for infertility. Again, we talked briefly about this as well. There are both male and female causes. We delved a little bit more in more detail to the sperm or male side of things, as we discussed in a previous lecture as well. On the face of it, it might seem that it's a 50-50 spread between male and female fertility. But you remember this big slice of the pie, which is idiopathic or unexplained infertility? The vast majority of these are thought to be, again, they're thought to be theorized. If we knew, it wouldn't be classified as unexplained, but they're thought to be predominantly because of a sperm issue, because on the face of it, everything else seems to be normal. So really, the purpose that we're trying to perform by characterizing the different forms of infertility, it's not some sort of sexist agenda, which unfortunately a lot of people do kind of tend to take the wrong ways, like, oh no, it's a male problem or it's more of a female problem. That's not the purpose behind trying to characterize what the main causative factor of infertility is. It's to try and find the best form of treatment. Okay, as you might appreciate, trying to treat a sperm problem is going to be very different from trying to treat a case of fallopian tube damage, or even trying to treat a case of fallopian tube damage will be very different from trying to treat a case of endometriosis, or indeed for that case, ovulatory failure. So, that's the main purpose for trying to find out what is the main causative factor behind fertility issues. So, as we talked about, the infertility itself, when groundbreaking decision about 10 years ago now, the WHO recognized it as a global disease. And because of this, or at least a large part of this, there's been a big uptick in terms of assisted reproductive technologies. You see, because while we talked about how different cases or different causes of infertility require different treatments, there are an equal number of many other treatments for each different type of infertility. So you'll have many different cases, many specific types of treatments to treat male infertility, and even larger to treat female infertility. There's also other more specialist methodologies as well, which we'll briefly discuss as we go along. But by and large, assisted reproductive technology, which is a very umbrella-like term, it doesn't refer to just one thing. It refers to multiple laboratory technologies together or collectively. And collectively, they're pretty good. They work quite well. And so infertility by itself is not necessarily as a quote-unquote death sentence. There is a treatment available for it, or there are multiple treatments available for it. So ART, collectively, they're very successful in treating infertility. They can cover or treat at least around about 70% of cases. Now, don't be confused in terms of successful treatment. What this figure just means is that ART can provide an avenue of treatment for about 70% of cases, which basically means this is excluding all the cases where unexplained infertility is a cause, because we don't really know where to start, you see. So ART provides, let's say, a ray of light, a ray of hope to be able to treat, at least provide a direction for some individuals who might not have any. And it's widely used. Like it or hate it, quite a lot of people are using it. It's becoming, instead of a last resort, it's now rapidly becoming a first resort for many more complex reasons, which we won't talk about. So more than 4 million babies have been born worldwide using these technologies. And as we discussed as well, about 7% of total births in some developed countries, we won't go talk about which ones, but these are specifically European countries. 7% of the entire birth rate of these countries began life in a quote unquote the test tube. So you see, whether you like it or not, this technology is very widely used. It's very successful. Okay, so it's in your interests and in many other people's interests as well to try and understand what makes this tick And that's what we're going to talk about today. We won't go into the details, the inner workings of it, of how each and every single methodology takes place. We'll go into that in later years. But it's more to give you an overview of what is the basic principle. What are we trying to achieve? What's the parameters one needs to keep in mind when trying to determine what type of treatment one needs to take into account. And really, what it's all about is trying to get these two gametes together. Okay, it's act of either fertilization or embryo development, or both within the test tube. So all, or pretty much the majority of assisted reproductive technologies, they surround, or their methodology surrounds, trying to introduce both gametes together, ensuring that fertilization happens, and then also taking it a step further and trying to select the best quality embryos. So it's also performing embryogenesis, or at least pre-implantation embryogenesis in vitro. So, when we talk about ART and we talk about IVF, as it's more commonly known, in vitro fertilization, we tend to think that there's it's a very modern technology. We've only just pioneered it in recent years, but that's not really true. If you think about it, as a species, humanity has been performing assisted reproduction, or has been assisting reproduction, let's put it that way, for a very, very long time. Think about selective breeding. Domestic dogs are the perfect example of humanity's selective or assisted breeding, where we have multiple species which arise from a few root species, from canines or from wolves, many hundreds and thousands of years ago, which we selected for certain characteristics or certain traits. But we have been assisting their reproduction. Think about cattle breeding. So, for example, in many different countries, prize bulls or prize horses, it's very, very big business to take their sperm and ship them all around the world to basically get the good genetics, as it were, especially for racehorses, for example. So all of these methodologies, where some form of assistance in reproduction is required, can be classified as assisted reproductive technology. And for that particular reason, you could actually go into the scientific literature, and at least for me, it probably could be earlier, but I was able to find the very first scientific report describing the first artificial insemination of dogs as early as 1783. So again, selective breeding. This, because it's assisted or artificial insemination, this is an ART. So ART has been around in some shape or form, as far as we've had an appreciation for breeding and technologies and all that kind of stuff. So it's been around with us for quite a while. But really, when we talk about ART, we're talking about the modern applications, in vitro, in a dish. That's the kind of space age technology that comes to our mind when we talk about IVF. So that very first modern application was performed in rabbits back in 1959. So 1959, 1958, very key experiments were performed using rabbits, using mice as mammalian models. And these were the very first descriptions of the ART technologies or IVF technologies that we are used to. And these were performed by this man, Professor Sir Robert Edwards. Now, it's not just Professor Sir Robert Edwards, but there was another clinician scientist, Patrick Steptoe, who was involved. We won't go into that too much, but by and large, Steptoe and Edwards were the pioneers. They were the very first ones to apply these technologies within humans. And back in 1978, this was successfully applied in humans, these technologies, to give us the very first IVF or test tube baby, Louise Brown. Okay, so this is way back when Louise was just born. And this is a more recent picture showing how gracefully Professor Sir Edwards, Sir Bob Edwards aged. And then here you have Louise Brown. And this is actually, I really like using this picture, and you'll see in a lot of my lectures and talks that I give that I'll use this picture quite a lot because it's very significant. Louise is actually pregnant with her first child in this image as well. So it's a kind of round circle, as it were. So these technologies since 1978 are very widely used now. And what was first applied by Patrick Steptoe and Bob Edwards in their initial experiments were routine, just in vitro fertilization experiments. But since then, many other technologies have come along. As we've improved our science, as we've improved our methodologies, as we've improved our understanding of reproduction, many different specialized methodologies have come about. But it doesn't really matter which methodology one uses. The basic principle or the basic goal will always remain the same. And you could try to split these out into 3 or 4 parts. Okay. Now, I say 3 or 4 because, again, this very much depends on what type of infertility one is trying to treat. So usually what would happen is you would begin with something called hormonal stimulation. Okay, now think of it, the reason why one would have hormonal stimulation is usually during the ovarian cycle, for a normal ovarian cycle, that is, assuming everything else is quote unquote normal, one would you would get 1 or 2 follicles every month that would mature, and they would rupture, they would undergo ovulation and release a mature secondary oocyte. However, this can be an issue when you're trying to do modern IVF because it's a numbers game, you see. The more oocytes you have to work with, the more embryos you can make. The more embryos you make, the more choice you have in terms of trying to choose the best one. And we'll talk about why you want to choose the best one in just a bit. But because in a normal scenario, what you really don't want to do is you don't want to put all of your eggs in one basket, as it were, at least put only one of your eggs in the same basket. You want to have as many as you can. So to try and get around that, there are regimes of hormonal stimulation which aren't very comfortable, I'm afraid, especially for the woman, because it involves regular injections of hormones or regular treatments of hormones. And what happens is that this overstimulation or increased stimulation, shouldn't say overstimulation, this increased stimulation, instead of having 1 or 2 follicles maturing, you can have around about 10 to 15 in an ideal scenario. Okay, but again, this isn't necessarily the all-encompassing thing. You might get cases where with the same treatment, one woman is getting maybe 15 oocytes being produced, but then others might only still be getting 1 or 2. So this is very much dependent on the type of fertility or the extent of the fertility that's being looked at. But by and large, this is the very beginning step. And this hormonal stimulation, it carries on into after the procedure as well to prepare the uterus for hopefully successful implantation. So while hormonal stimulation happens, and once the fruit is quote unquote ripe to pick, the next thing that one needs to do, the very first step in terms of basic principles, is that one needs to isolate gametes. Okay, so we need to get an egg or an oocyte, and we also need to get sperm. Now, once we've isolated these, again, there are many different technologies to do that. By and large, it's much easier to isolate sperm than it is with oocytes. But again, dependent on the type of infertility, for example, with azoospermia, again, it's probably going to be easier to get the oocyte than it is going to get the sperm, because you actually don't have any sperm production. So the very first thing, or the very first step one needs to do, is to isolate the gametes. And again, this will differ depending on the type of infertility. Once you have a strategy for isolating the gametes, these then need to be combined. Step number 2 is basically mixing the egg and the sperm and then crossing your fingers and making dua, because that's all that we can do. And hopefully fertilization will happen. Now, again, it's not quite that simple. It very much depends on the type of infertility, because there are also many other types of treatments specifically tailor-made to facilitate this process. And we'll talk about 2 of them in just a bit. So once step 2 is complete, hopefully you should be able to get a mature embryo. And once you get your embryo, we need to transfer these embryos at the blastocyst stage into the uterus. Now, you see, at this stage, in the past, people have transferred historically multiple oocytes or embryos at the same time. I should say embryos, not oocytes. And the reason for this has always been to try to increase the chances of a successful pregnancy. So once IVF first started, it was routine to transfer 3 to 4, maybe even sometimes 5 embryos into the uterus and hope at least one of them will stick. But you see, recent research has suggested that the more embryos that are transferred into the uterus, the lower the chances there are for even one embryo to be able to successfully implant. Because you see, at this stage, these embryos, they're multicellular organisms that will be competing with each other for limited nutrients within the uterus. So for many reasons and others, then you have, risks to the risk to the embryos themselves. You have risks to the mother as well. With the more embryos you have implanted, the riskier this pregnancy becomes for everyone involved. So a lot of recent research has suggested that really we should try to minimize the amount of embryos that we transfer. So nowadays it's more common to transfer two, but the ideal scenario is a single embryo transfer. Now, because we want to transfer one single embryo, or at least limit the number of embryos we're transferring, we need some way to choose how to select the best crop out of these 15 embryos that we have. If we're so lucky, sometimes, or at least most of the time, you won't even have that choice. You'll only be able to generate 2 or 3 embryos, and you just transfer everything just and cross your fingers. But for this, there are other methodologies which are specifically designed to monitor and assess the quality of the embryo itself. And usually this involves time-lapse imaging, and where essentially you make movies of the developing embryo and you decide based on these certain parameters. But you see, it's not just the embryos that we have to try and decide is good or not, quote unquote good. Because we can't just use every single gamete that we have. If we use a bad quality gamete, then of course the embryo is going to be a bad quality as well. So remember with the sperm, we talked about definitely specifically with the sperm, not all sperm is capable of even fertilization, let alone embryogenesis. So only it's estimated about maybe 1 out of 100 sperm even have the capability in an ejaculate of being able to reach the oocyte, let alone fertilization. So we need first, once we've collected our gametes, to try and assess which ones we want to work with, at least try to sort out the good from the bad, especially in the case of the sperm. With the oocytes, of course, it's relatively easier, but that much more harder as well, because you need to be very strict. Of course, at this stage, there's not much that we can do, you see. If we try to do any sort of invasive tests on these gametes, then we have a very real danger, very high danger of damaging these gametes. So really, all that we can do is use a microscope and basically just come up with different parameters to look at using a microscope, to look at these gametes. So because of that, there are certain criteria that have been, let's say, found out or agreed upon based on certain morphological characteristics. And we'll talk about that in just a bit. So this slide is just another summary that I wanted to just put into context for you in terms of the hormonal stimulation. So in a normal ovarian cycle, you're going to be getting your primordial follicles, and they're going to start developing with the primary oocytes. At ovulation, you're going to get the secondary oocyte that's being released that will then go for fertilization. So the hormonal stimulation itself, that's required to increase the number of eggs obtained. So the more eggs you have, the more oocytes you have, the higher your chances of success. So because we only have the microscope really at our disposal to be able to do anything, all we can do is just essentially look at our cells and be able to make some sort of, sorry, I just looking at the chat and I see someone is talking about genetic screening. Hold your horses, we'll talk about that in just a little bit, okay? We have a solution for that as well, but just bear with me. So because we only have the microscope with us, it's really a beauty contest, you see, at least as far as the oocytes or the eggs are concerned, because we can't really do anything else. But you see, because we only have the microscope, and because there's so many of these procedures that are being done, and because a lot of people are doing this with just experience and different studies and the intensity and depth of the studies that we've had, many different morphological criteria have been placed which correlate to successful fertility. Okay, so again, with oocytes, this is going to be everything that you see. It's going to be the cumulus cell layer, the density of it, the shape of it. We're talking about the zona pellucida, the shape of this as well, whether or not it looks nice. So this is a very good example of a very healthy looking human oocyte. So you can see it's a very nice and thick zona pellucida. You can see a very well-defined plasma membrane for the oocyte itself. You can see here the first polar body. If you look at the cytoplasm, it's very clear. Okay, there's no indentations, there's no kind of craters in the middle. It looks overall a beautiful and healthy looking oocyte. Okay, and there's also a very nice association between the zona pellucida and the ulema over here. Now, I might sound like I'm having a bit of a geek out on this, but you see, all of these things seem to be very, very important factors when you consider what gametes can be used for successful treatment. The same thing is also applied for the sperm, but you see with the sperm, because there's many, it's a sheer difference in terms of scale, you can afford to be a little bit more invasive with this in the vast majority, of course, of cases. In cases where you have very extensive azoospermia, you actually have a much harder time compared to the oocytes as well. So the kind of things that we're looking at the concentration, so the number of sperm. That's a very good indication of whether a good sample is present. If you have a very low number of sperm, then you know that even the sperm that you have, chances are they're not going to be very good. We're looking at the motility. So this is essentially, as we talked about, looking how well the sperm swim. So if you have a lot of sperm, but they're not very motile, that just means that they're dead, right? So there's not going to be much that you're going to be able to do. But that also then determines the course of treatment that you're going to have. We'll talk about that in just a bit. Again, with the morphology, same criteria with the sperm. You're looking at the shape of the sperm head. You're looking at the clarity of the cytoplasm. We talked about this briefly as well. You're in the previous lecture, we're looking at the midpiece. You're looking at the shape of the tail, all of these types of things. And again, because there's a difference in numbers in any kind of routine sample, there's also other criteria that are a bit more invasive, but you use these tests only if you can afford to take a certain number of sperm. And one of these is something called a viability test. And essentially what it is, you take a little bit of sperm and you add it to a dye. And the idea is that if you have holes in your membrane, i.e. if your sperm is damaged, then the dye will be able to enter the sperm head, and you'll be able to stain the sperm head. So if you have a lot of sperm which are being stained, that means that a very large proportion of your sperm are non-viable. They have holes in their plasma membrane. So that correlates to low chances of success. Another criteria, which is more experimental, but it's really rapidly becoming a very increasing indication of fertility prediction. And this is something called DNA damage. So think back to your spermatogenesis lectures. Think about how the nucleus is condensed. You have histones, which are replaced by proteins to get it to be a very, very condensed state, even more so than in a normal cell. And what can sometimes happen through various different things, we won't talk about what these things are, but you can get bits and pieces of DNA being damaged, okay? Either by UV or oxidation or just throughout the process of packaging itself, the DNA can be shred up into bits and pieces. And of course, if the DNA isn't packaged properly, then that defeats the purpose of that particular gamete. So this criteria is also increasingly being used to assess DNA, to assess sperm fertility rather. So based on these criteria, then, one will be able to decode or decide what course of treatment one needs to use. So I mentioned briefly this umbrella term called in vitro fertilization. Now, think about it. In vitro is Latin for inside a dish, okay, or in a tube. So all methods of assisted reproductive technology can be classified as IVF, especially if there is a fertilization event that's involved. So the vast majority of ARTs, various technologies, various methodologies, because you're performing all of these methods in vitro, they can be classified as in vitro fertilization. So that's the general term that the public uses. Now for us though, as budding scientists and clinicians, because we're learning more about this in detail, IVF refers to a particular type of methodology, okay, at least within the field. And this is the more common type, where you essentially take an oocyte, take some sperm, and you mix them together. Okay, so over here, this is an example of a human IVF that's being done. In one drop, you have 1 or 2 oocytes. Most of the time, it's 1. And then here you have a smear or a droplet of sperm. And essentially what happens is that the embryologist will combine these two, not completely, but by using a single line or a streak connecting these 2 lines. And the idea is that sperm from this membrane will flow into the drop of this oocyte and hopefully be able to make their way and fertilize the oocyte itself. So for many years, many years, this was the default methodology, okay? A lot of clinicians preferred using this. This was the most sophisticated technology that one wanted to use. But of course, this has its own limits. If there's not enough sperm, that decreases the chances of successful fertilization. If the sperm isn't motile, that also, there's no point in this, the sperm won't be able to swim towards the oocyte. If the morphology isn't correct, or if the morphology is defective, and I believe I was asked a question about this, defective morphology of the sperm, essentially that indicates that the structure of the plasma membrane of the sperm head is defective. So all of the receptors that are required for the sperm to bind with the oocyte or recognize the oocyte will most likely be defective as well. So an abnormal morphology, think back to the previous slide, we talked about globozoospermia. Yeah, that round-headed sperm, that balloon on a string morphology, that's a very abnormal morphology of sperm. There, you could also have cases where you don't have any acrosome. If you don't have acrosomes, then you don't have the digestive enzymes required to remove the cumulus cells and the zona pellucida. So that, again, results in a failure of fertilization. So what happens if you need a more extreme intervention? Okay, what happens if this normal method of just mixing the two gametes and hoping they fertilize, you need something more invasive? That's where a more recent technology has come in. It's called intracytoplasmic sperm injection, also known as ICSI. And this is a very powerful technique. Okay, it's also the kind of space age science, at least I was promised as a young student. It involves this really complicated looking setup, which is essentially a microscope with two injecting needles. Okay, now I should mention that everything here, including this fire hydrant and this CO2 tank and this operator, all of these things, including these empty plugs, are required to make this work. Okay, so not very straightforward, very complex piece of kit. It's a specialized microscope as well. But the business end you see is here. Inside this dish, you have two different drops, three sometimes, depending on which methodology you're using, but you have drops on your dish. You have a sperm drop and you have an oocyte drop. And you have two different needles, okay? On the one side, you have a much larger needle, which is called the holding needle. And this latches on and holds on to the oocyte as such. And on the other hand, you have an injecting needle. And what this does is that it takes up a single sperm. Okay, so the operator using this is able to select a sperm which looks nice. It's exhibiting good morphology and motility, and it's picked up into this, and a single sperm is injected into the oocyte. So if you bear with me, I have a movie that I want to show you of this methodology in action. There you go. Okay, so here you have the injection pipettes. There's a sperm injected inside. So it's aspirated a little bit to create room for the sperm. There you go. You burst the plasma membrane and the sperm is deposited right in. And this is then removed. So we just loop this again just to play this for you. So you see, this is a very powerful method, okay? Whereby the sperm is delivered directly within the oocyte itself. And this treats a huge range of predominantly male fertility issues. So think about it. If your morphology is abnormal, if your motility is abnormal, if your concentration is abnormal, even in terms of female infertility, where you don't have that many oocytes to work with, and you want to ensure that successful fertilization occurs, this method is used very routinely now. It's by and large the most popular method of ART. And it's very fast becoming the very first port of call. And what that means is that very routinely now clinics, they don't even care about what the particular fertility issue is. They just use ICSI by default. Now there are issues with that, and we'll talk about that briefly. But that just indicates to you just quite how powerful this methodology is, and that it's very rapidly becoming, since its first inception back in 1992, becoming the first port of call, the very first default treatment that's used. But that's not the only type you see. These are the two major, IVF and ICSI are the major fertilization assisting methodologies to create an embryo. But ART is by and large not limited to just those two. In fact, you don't even need just those two to be able to perform a lot of these other types of methodologies as well. So one thing, again, to combat more female infertility is something that's been developed called in vitro maturation, where instead of obtaining secondary oocytes or ovulated oocytes, increasingly what is now available is something called in vitro maturation, where immature primary oocytes, also known as at the germinal vesicle stage. So remember, at the primary oocyte level, these oocytes are arrested at the first mesophase. Sorry, at the first meiosis, not mesophase, it's prophase. And these are obtained from the immature follicles, and they're matured, not in follicles, but inside a dish, in specific media. So you go from primary to secondary oocytes within a dish itself. And this, this has many advantages. This gets around hormonal issues, of course. So hormonal overstimulation is a bit of an issue where some women can suffer from ovarian hyperstimulation syndrome, where too much hormone is delivered for that particular female. And that leads to many other conditions, which I won't talk about for this lecture. But this kind of gets around it. And it also increases the number of cells that you can work with. You can go up to even 50. If you can find the follicles, you can remove the oocytes from those follicles and mature them in vitro. You also have cryopreservation or vitrification, which is essentially snap freezing your gametes. So this is usually used in fertility preservation. Now, it's not just fertility preservation. This also gives you, let's say, chances at multiple rounds of treatment. Okay, and we'll talk about why you would need to do that. But you could just harvest gametes at one time and then freeze all of them in liquid nitrogen and even much more colder substances, which we're now trying to get around in terms of research at this stage. And this, if you can harvest them just at one time, you preserve them, and then you can use them at multiple times. And you can modify which treatments you want to use by trial and error later on. But also in terms of preservation for cancer survivors, also for fertility treatments later on for, let's say, transplantation issues. On the more diagnostic side of things, you also have something called embryo biopsy. Now, this is a really cool piece of technology where essentially you take a blastocyst, you take the embryo, or even before even the morula stage, at the 6 or 8 cell or the 12 cell stage, and using a laser, you digest away the zona pellucida and you cut off either a little bit of the blastocyst, or you take out one of the blastomeres or one of the cells from inside. And then using this biopsied material, you can then perform genetic tests. And these genetic tests, they can be either prenatal genetic screening or prenatal genetic diagnosis, okay, PGS or PGD. Now, these are very, very useful, you see, predominantly, especially in terms of fertility preservation, because it's found that as females age, oocytes from these females undergo something called aneuploidy, or abnormal number of chromosomes, where essentially the meiosis during the maturation isn't normal. So you have either more or less numbers of chromosomes, from either more or less than 23 chromosomes present within this oocyte. And of course, what this would result in, you would get normal fertilization, you would get a pre-implantation embryo up until the blastocyst stage, but this will not be a viable baby. It will not be able to develop into a viable baby. So using these techniques, for older women, one can try and select the more normal embryos and only transfer those. But then, of course, you also have genetic diseases where embryos can be tested to see whether or not, especially in the case of if both the male and the female are carriers for a certain disease or genetic disease, then you could see whether or not this embryo is afflicted. You could also see whether or not this embryo is even a carrier of the disease, performing these genetic tests. So very, very powerful range, or very powerful suite of methodologies that one could use. But we've just talked about up until now how wonderful this technology is and how cool the techniques are. But you see, we're just in this final section. It's not all roses and daisies, okay? It's a very powerful set of methodologies, but it's not so straightforward because there are very, very much issues with ART. Numerous issues, we should say. The very first of these, remember I showed you this picture and I've just showed you from a different angle. This just kind of gives you an idea with ICSI, for example, just how complex everything is. This person is not only responsible for performing the procedure, obtaining the gametes, looking after the gametes, looking after the embryo, but also responsible to be the technician which looks after this microscope, this entire setup. So as you can appreciate, not everyone will be able to do this. Extended training is required, specific training is required as well. Now, the problem is that not everyone has the same set of standards. Okay, European standards are different from American standards. GCC standards are very different from, let's say, India or Pakistani standards. Around the world, there isn't one set of standards that one adheres to. The WHO tries to give some sort of guideline, but again, as we've seen with the ongoing pandemic, not everyone tends to listen to the WHO, unfortunately. So this is one of the first obstacles, let's say, is that it's so highly specialized and so requiring of extended training that it's very difficult to establish in the first place. Not everyone can do it. And even though ART can be used to treat about 70% cases of infertility, the success rate has a lot of room for improvement. Now, globally, birth rates in terms of the success rate in terms of how many babies are born for each round of ART, which means hormonal treatment, gamete selection, fertilization, and embryo transfer, that's one round, they rarely exceed 30% for each cycle or for each round. Now, considering that not only are you bringing the horse to the well, you're also opening its mouth and pouring the water inside with these methods. And yet, for IVF, success rates rarely exceed 22%. Same thing for ICSI. Even especially with this one, you're taking a sperm and injecting it directly into the oocytes. Despite that, despite this level of invasion, it is successful. But for each round, it's only about 23%. To be able to get a successful live birth, more than one round is usually required. So not just one round of treatment, but at least 2 to 3 times before a successful pregnancy can be established. And live birth is something completely different. That could take even up to 5 to 6 times, but that very much depends. And these figures, they vary throughout the world. So again, there's another lack of global standardization here as well. Now, it's not just, let's say, logistical or technical issues with ART. More worryingly, ART is now being associated. Now, we have to be very careful here. I'm not saying that ART is causing these diseases, but ART is increasingly being associated with both genetic and epigenetic conditions of disease. Now, genetics, it's very straightforward. If one's infertility is due to a genetic deficiency, then, if you're doing ART, the chances are that your child will also have the same genetic deficiency that you are creating. So in actually what would happen in the gene pool, if you're unable to reproduce because of a certain genetic condition, your genes would no longer be passed on. So that problem would be removed from the gene pool, as it were. But because you're doing this problem, ART, you're getting a successful childbirth, then these genetic issues will persist within the population. But that's okay. That's something that can be managed, okay? Because, well, does it matter if the genetic conditions are around? You were able to benefit from ART, and I don't mean anyone here per se, I just mean, they're thinking out loud. That one was able to benefit from ART, so that same option would be available to your children as well. So that genetic condition no longer becomes an issue. But the more worrying thing is epigenetic conditions. Now, what I mean to say here is that an increased number of babies born via ART seem to have or are associated with higher proportion of these particular epigenetic conditions. And these include Beckwith-Wiedemann syndrome, you have Angelman syndrome, and you also have Prader-Willi syndrome. Now, all three of these, I won't go into what these diseases are, and it would be nice if you could remember just the names of the syndromes which are associated with ART, because these three are the major ones that are associated with them. But again, it's not really clear whether ART is causing these, or whether, because it's a smaller population, whether or not it's similar to what you would see in a normal global scenario to begin with, because not everyone will be declaring or following up all of the children that are born by Beckwith-Wiedemann or Angelman or Prader-Willi syndromes, at least to the same degree as ART babies would be. So it's not really clear, but we just can say that a higher proportion of babies seem to have some sort of epigenetic abnormality. Now, the reason why it's not really so sure is because you see, at the moment, the very first IVF baby, Louise Brown, was born in 1978. So let's round it up, 1980, right? So it's only really been about 40 years since this technology has been around. Okay. Now, yes, it is Nobel Prize winning work. So Patrick Steptoe and Bob Edwards, they won the Nobel Prize in medicine for this work. So it's come a really long way, but we're still in the infancy of this. Okay. So all of the IVF babies that were born, the first round of IVF babies, they're only just now kind of reaching sexual maturity and trying to have children of their own. So it's now is the time that we're actually beginning to study the effects, the generational effects and intergenerational effects of ART. And unfortunately, what we're finding, it doesn't really offer much hope, you see, because a lot of studies, especially as far as male fertility is concerned, are indicating that there are long-term effects in terms of genetics. Now, this in itself is a little bit confusing because one could use this argument either way, depending on which side of the fence you sit in terms of whether or not one should use ART or not. But what the studies show is that over the past 60 years, at least in developed countries where these figures are available, there has been around about a 60% decline in sperm quality. Okay, so what this basically means is that male parameters seem to have fallen by about 60% over the past 60 years. Now, before you become all alarmed, there are many other factors to this. So these are data that are taken from fertility clinics. So males from these fertility clinics, the higher chances are would be they're already suffering from some form of male fertility issues. So does it mean that instead of the global population of males becoming less fertile, instead of that, is it just the males who are attending fertility clinics are getting more infertile? Don't really know. Okay, it could be that you're seeing this intergenerational effect where multiple generations of the same family, of different families, are attending clinics, and this genetic issues are becoming more and more predominant. Now, I've come across such issues myself, where in clinics, I won't say where, but it was a GCC country, where you had 3 generations of the same family attending the same clinic for their fertility treatment, and this seems to be becoming more and more common. So could this be an issue that's leading to a persistence of the problem? Kind of like the tobacco industry, you hook them while they're young, and this is as young as you can get. But just to quickly conclude, even though it's not the perfect solution, there is a lot of room for improvement, and we are improving these technologies every single day. There are multiple publications. We make advances every single day, and our understanding of reproductive biology improves, our treatment methodology improves as well. We kind of realize that whether we like it or not, whether or not it's successful, whether or not there are ethical concerns, whether or not you agree or disagree with ART, it's here to stay. Okay, it's the final call for hope for many infertile couples. And like I said, for the vast majority of cases, it is able to result in success. 4 million babies born worldwide. The real figure is probably much higher now. These are a little bit old figures. 7 to 10% of total birth rate. So, it's really, really working. So really, I think as budding clinicians yourselves, you also have a kind of, let's say, moral and professional duty to understand the technology of this. Okay, it falls down to us to educate people about this, particularly in areas where infertility is still viewed of as a taboo. Now, the main purpose of this talk also was to make you aware of the types of treatments that are available. Okay, now use the common cold as an analogy. You have if you have the common cold, there are multiple remedies that you could use, both from your grandparents as well as from the pharmacy. So view infertility as the same kind of thing. Okay, there are treatments that are available. They're not perfect, but they're becoming improved more and more as we go along. Okay, so hopefully this has been very illustrating for you. And if there are any questions, I guess I could take them now. Let me just see if there's people have left anything in the chat. So we fertilize more than one oocyte and choose the best embryo. Yes, with hopefully if we can afford that. And yeah, I think you assess the eggs and sperm before fertilization, assess the fertilized egg and embryo. Well, you're doing assessments at all stages. So you're assessing the oocytes, you're assessing the sperm, you're also assessing the event of fertilization. You're also assessing increasingly now the fertilized zygotes and development throughout embryogenesis. So the entire process is you have multiple points of assessment as you go along. So there is another way, which is one oocyte, one sperm, one embryo. Yes, theoretically, that's what we want to try and achieve. But practically, this is near impossible, unfortunately, because, you know, if you're only able to get one oocyte, even after hormonal treatment, then that probably suggests that there is a much larger issue at play. And if you have just lower number of oocytes, you're not going to have successful fertilization, unfortunately. So I think you collect multiple eggs, multiple sperm, analyze. Yes, exactly. Yes, I can confirm that. What makes ICSI different to IVF? Remember that IVF is an umbrella terminology for as far as the general public is concerned. Both ICSI and IVF are types of in vitro fertilization, but as far as we're concerned, IVF is where you mix the gametes together. So you connecting the two droplets, sperm swims. ICSI is where you take a single sperm and you inject it directly into the oocyte. So you can use it to treat much more specific male factor conditions. Won't the oocyte get damaged? Yes, it will get damaged, especially with ICSI. And not just with ICSI, though, with many different types. Sometimes you have the biopsy, you have the aspiration, you have the removal of the cumulus cells. So it's just the fact that it's IVF, it's a very invasive set of methodologies. So that's why you need to monitor it. But you know, I suppose given the outcome will be a successful birth, then a little bit of damage can be managed. And that's what we're trying to do as a field now, to try and limit these damaging events. So if in vitro maturation, who will prepare the uterus and other things prepared by the follicular hormones? Again, this will be hormonal treatments. Hormonal treatment is quite common for rounds of IVF, and what type of hormonal treatment you do very much depends on the treatment regime. So if you're doing undergoing a round of in vitro maturation, then you will have a specific hormonal treatment. If you're just doing IVF or ICSI, you'll have a different hormonal treatment. Anything about genetic engineering? We don't have enough time to talk about this loaded issue, but genetic engineering for human embryos equals to bad. Just remember that. So doctor, can you explain in vitro maturation? So in vitro maturation is where instead of doing a hormonal treatment and getting multiple oocytes at the secondary oocyte stage, so mature oocytes, what you're doing is that you're limiting the amount of hormonal treatment. So you're not maturing multiple follicles, but you are taking immature primary oocytes from immature follicles, you're taking them outside of the ovary, and you culture them in vitro, and you mature them using different media. So that's what in vitro maturation is. So according to what we choose the appropriate tech, we choose the appropriate technology based on the type of infertility. Okay, so for example, if you have a low sperm count and zero sperm motility, then you won't be doing IVF. The most appropriate technology will be ICSI. So yes, a number of people are asking me about their own interest. Yeah, if you have any specific questions, please feel free to email me at any time, and I'll try to get back to you as quick as I can. So for carrying on from the previous explanation, for example, if you have issues with obtaining oocytes, so for example, if one needs to do in vitro maturation, or even after hormonal treatment, you're only getting 1 or 2 oocytes, then you don't really want to take a chance of doing IVF, because you want to be in a bit more control. So even even in certain female cases, or cases with female fertility, one would want to use ICSI as well. Okay, so it's the type of technology or the type of methodology that you use very much depends on the type of infertility. So to be able to understand ART, we need to understand what causes, or what is the basic cause behind fertility, which brings us back to those figures that I was showing you. So it's not a boys versus girls competition, by the way, which boys unfortunately lose, because as I mentioned to you before, the vast majority of cases, the leading causative factors are a male issue and not female. But it's not a boys versus girls competition. It's very much to try and understand what methodology to use. Anyone have any other questions?
Thank you so much, Doctor.
That's wonderful. So yes, thank you very much, everyone, for listening. Again, if you have questions, just feel free to email me. If not, all the best of luck. Hope you stay safe, and inshallah, see you soon.